This YouTube thing is interesante. All right, looks like we got some folks here already. Hi, Surya. Hello, Parth. Hi, Ronit. I'm doing well. Thank you very much. How are you doing? How's everybody doing? I started just a little early just to make sure that I get set up properly before we get started with questions. And hello, Shreya. I love how as you're all rolling in, you're saying hello. So polite. Hey, John, I'm doing well. How are you? It was nice chatting with you in class today. Hi, Anika. All right, everybody who's here. It hasn't, the session hasn't started just yet. I just wanted to start just a tad early just so I get set up. Hey, Karthi. Hey, Stefan. Nice to see all of you pop up. So those of you who are here already, I was thinking of doing this one slightly differently. I'm thinking maybe we should try to cover one topic at a time instead of bouncing back and forth. So like, we'll start with photosynthesis, and then we'll go to plants, then we'll go to cell respiration and things like that. What do you all think? How does that sound? Hey, Shreya. Hey, Shrika. Yep, Ronit, you are right. Those are the main topics that are covered on the test. Who else has shown up? Hey, Arnav. Nice to see you. Hi, Ishida. Yeah? Chaitra? That sounds good. Yeah, I agree with you, John. I think anyone who joins will ask random questions, but that's okay. We can just remind them that we'll get to that, or we've already gotten to it, and then at the end I can go back over stuff that we've already covered. Shreya, you think that sounds good? All right, we are almost there, almost ready to go. Ronit, 23 people. Is that how many people we have right now? I don't even know how to check that, so thank you for telling me about that. Where do I see that there's 23 of you? I imagine we'll have more people. Oh, I see it. I see it. It says 22 people watching. All right. Okay, it is 6 o'clock. So let's go ahead and get started. So I wanted to, how for every review session, live review session, I've been starting it with some kind of food as the theme. So I have two things today as the theme. So last time I had a pomegranate. This time I have something related to that. I got pomegranate juice. This stuff is amazing. And check out this glass mug that a student got me a couple years ago. It says, "I'm not saying I'm Batman. I'm just saying no one's ever seen me and Batman in the same room." So that is the food I wanted to start with. And the other thing that I wanted to show you, the other thing I'm going to be snacking on as we are studying together. Ronit, no, it is not homemade. I do not make my own pomegranate juice. That would take a lot of effort. And pomegranates aren't cheap. And the other food that I want to share with you all, well, I should share it with you, but grapes. I'm snacking on some grapes tonight because I find it to be a super healthy snack. And as you all know, I love fruit. And so, yeah, it's going to give me that glucose that I need. We're talking about photosynthesis, so I'm going to talk about the grapes as we're having conversations about it. Because I do want you to know where the sugar in the grape came from and where it was made. Anika, they are not cotton candy grapes. Anika, you and I shared some cotton candy grapes, didn't we? I think you and I and Jenna had some cotton candy grapes. Who else has shown up? Hey, Nitin. Hey, Gondar. It's nice to see you all. Jasmita, my TAs were telling me today that you are one of the most honest students they've ever met. And I think that's awesome. And I agree. Let's see, Ronit, will the test be in the same format as the past test? Yes, it'll be the same format. It'll be C-level questions, B-level questions. I think there's something like 22 C-levels, something like 11 B-levels, and then 2 A-level questions. Hey, Arjun. Let's see, Ishida, like plants and bacteria dust fermentation. Yeah, we're gonna talk about that too. I'm not this right here, by the way. This is not fermented. This looks like wine, but it is not. I enjoy the taste of pomegranate juice much more than I enjoy wine. My wife would disagree with that comment. Hmm?
I don't disagree with what you say.
But for yourself.
Yeah.
Yeah, she's listening in on this.
I'm not.
All right. Let's see. John, you stole some of Jenna's cotton candy grapes. I hope you enjoyed it. The cotton candy grapes were amazing. I actually went back to that store for more, but they didn't have any. I was gonna get more to share with you all, but they're out. They're a popular item. So next time they come around, I'll get us some. Shreya, you want to try cotton candy grapes? Shreya, yeah, I think it was 5th period where I was eating cotton candy grapes and we ended up sharing it with each other. Chaitra, same. Jenna, yeah, Jenna, you remember those grapes? Rana, do you want to see my dogs? I don't know where Kuchuk is. I think Dakota is laying behind me. I can call him over in a little bit. Hey, Jacob. Jenna, you think that was a long time ago? That was 2 months ago. That was not a long time ago. John, no, this is a different Jenna. Yeah. Hi, John. Arjun. Yep. I'll see if I can get my dogs over here. Gandhar, what do you have 5th period? That's when you should have come in. We would have shared some grapes with you. Everyone wants to see my dogs. I was telling 3rd period today that I'm going to try to have a study session before finals week where I bring in my dogs, but we're technically not supposed to have dogs on campus. So I don't know. It would be awesome if you could have a study session at a park or something, but with December weather, you never know. So I'll think about that one. Ishida, where can you find these? You mean the cotton candy grapes? Trader Joe's carries them, but they go quickly. You have to be on top of it. Like you have to show up as soon as they open, and basically, like I think you can reserve them too. You can reserve, I forget, as many as so and so, like batches or boxes. But yeah, people are all about those. Hey Ria, nice to see you. Gondar, what is it you want to see? Show me. I don't know what you want to see. What's up, Sai? Nice to see you. Gondar, you have 5th period Java, or what I pronounce Java. Run it. Yeah, you have Java as well. Like a session before finals. Yeah, Ishida, that would be awesome. I think that would be really cool if you have that before finals. I totally agree. Yeah, I know we're not asking bio questions, but that's awesome. We'll get to bio questions in just a moment. We're talking life right now, homies. It's even more important than bio. Explain apoptosis. Yeah. Arjun and I will get to those. Are we starting? Yes, we are going to start. Let's get started. Hi, everybody who's rolling in. Archit, Ruhi. Hi, Jonathan. All right. Okay. Ron at Real Talk. Okay, so let's get started. I'm thinking we start with, how about we start with photosynthesis? Let's have that be our first business that we talk about. So regarding photosynthesis, I want you to remember the equation or the reaction for photosynthesis, right? What do you need? And this is something you're going to need to know for tomorrow. So what are the reactants and what are the products of photosynthesis? So we're going to need sunlight. Remember, the sunlight can go on the arrow or it can go in the reactants. We're going to need water and we're going to need CO2. And what are our products? Remember that? We're going to get oxygen and we're going to get glucose. How many glucose molecules do we get per round of photosynthesis? 1. We get 1 glucose molecule. And how many oxygens do we get? We did this in class. So I would say if you are watching this and you're just looking at your notes or trying to study at the same time, take out your notes for photosynthesis and let's look through those. Okay. Chaitra, I don't have my whiteboard, but I can go look for it if that helps. It's in there. Okay, my wife's getting it for me. Thanks, son. So let's see. Yeah, no, you don't need it. If we might need it, so we'll see. So basically with photosynthesis, I think, yeah, thank you. Thanks, son. So I know that not every teacher does LED electron. 6H2O. Ishida, yeah. I'm glad that you guys, yeah, it looks like you're looking up what goes in, what comes out, the reactants of the product. So yeah, so I know that not every teacher does LED electron. So some teachers get really sort of deep into it, and some haven't. I'm one of the teachers who sort of dove deep into it because that's just kind of Eddie allows us to do that through that story. So I posted Eddie the Electron story on Google Classroom, so make sure you check that out. And those of you who haven't heard the story, that might actually be helpful if it's something that your teacher wants the details of it, basically. Yeah, Gondar, I think you got Eddie too, right? Eddie's such a great story. Ishida, I totally agree. Eddie is a great story. I love Eddie. We've considered over the last couple of years not teaching photosynthesis as detailed as we do. And so, and I think it makes sense why some teachers don't teach photosynthesis to that degree, because it's no longer part of California standards in the way that it used to be. But I think I hear from students who are no longer bio students. They took it several years ago, even college students who have bio with me. And, you know, when I ask them, tell me a couple things you remember, Eddie always comes up. And so I've had a hard time letting Eddie go because I feel like it's a really nice way to teach photosynthesis. So that's the deal behind that. Regarding glucose, remember it's C6H12O6. Remember that. And remember that in photosynthesis, you're going to have two different reactions. You're going to have light-dependent reactions, and then you're going to have what's called a Calvin cycle. Again, if in your class you did not cover these things, then don't worry about them. You can just kind of hang out with us right now. But those are the things that we want to know. Remember light-dependent reactions. Remember there's a ETC, electron transport chain. Remember that sunlight excites electrons. Electrons go down to ETC. While they're going down to ETC, remember they meet ADP, a depressed person. They turn them into ATP, a thrilled person. And then from there, you are going to get an electron carrier, NADP+. That's going to grab electrons. It's going to put on a hydrogen, and it's going to take the electrons to the stroma, not the stomata. It's going to take them to the stroma, and then that's where the Calvin cycle takes place. All right. Remember that Eddie was first in the thylakoid discs, and that's in the grana, and that whole thing is in the chloroplast. All right. Let's see. Let's see if there are any questions about that. So ask me any photosynthesis questions you got, because right now we're discussing photosynthesis. So what you got? Can you explain some more about the structure and function of palisade and spongy mesophyll layers? Sai, so honestly, I'm not going to be too concerned about you being able to kind of figure out what's where. I think I just want basics about the palisade and spongy mesophyll. I think just the basics of the worksheet that I gave you about leaf structure, that's what I'm looking for. I don't want anything extensive. Are we going to need to know about sources and sinks in a plant? So Arnav, ideally, I wanted to teach that to you all, but we didn't have time for it. So I know it was in one of the worksheets, but you don't need to worry about that. So everybody, do not worry about sources and sinks. It's a really cool concept. I would have loved to teach it to you, but we ran out of time. So yeah, Gondar, we did ADP, a depressed person. It's pretty awesome. And then ATP, a thrilled person. I did not come up with those. I wish I had. I think it was a former TA who came up with them. Yeah. Difference between stem cells and cancer cells. So we'll get to that, Archit. I think we're just going to do photosynthesis for a little while, see if anybody has questions about that. So Surya, transpiration is evaporation of water from plants. So I want you to remember that basically the water goes up the xylem in a plant. And there are three processes that are actually helping. There's adhesion, which is water holding onto the sides of the xylem. There's cohesion, water molecules holding onto each other. And then there's transpiration, which is like a vacuum from up top that's pulling the water up. So those three things all together are going to help. And they're all together, they're called capillary action. And that's what's pulling the water up a plant. Let's see, I'm falling a little behind with the comments. How do plants come from the air around them? Okay, so John, so this is awesome. I love this question. So plants, when you look at a plant, when you touch a plant, when you touch the leaf of a plant, when you touch the stem of a plant, you ask people like, where does this come from? And there was I think, a study at Yale or Harvard, I think when bio majors were graduating, I don't know if it was bio majors or just general students graduating, they were asking them, where does the substance in a plant come from? And a lot of students were saying things like water or soil. And that's not true. It's actually CO2. So it's taking it in gas form, and then that gets turned into carbon. And so that's where it comes from. What are the basics of the palisade and spongy mesophils? Let's see. Let me come back to that one. What are the limiting factors for plants in a jungle? What about a desert? Around it. So really, I would say sunlight is a really big deal. One of the reasons why you see a lot of plants growing taller and trying to, their canopies like way up here is because they're trying to reach for sunlight. So like I have a tree in my backyard. It's a maple tree. And this maple tree has made all kinds of weird sort of like curves as it's going up. And the reason for that is because it's constantly trying to look for sunlight because there was another taller tree in the way. Cool. Factors for plants. What else? What does capillary action have to do with water traveling up the root of a plant? Capillary action, Ria, is basically when you get transpiration, adhesion, and cohesion all going through the stem. So remember what we talked about in class about like when you buy flowers, they tell you to cut an inch or two from the bottom of the stem. And why you have to do that is because all those three processes are capillary action, and that's what's pushing water up the stem. Jacob, can you tell us what the topics are on the test? So Jacob, we have someone who made we have a student who made a really cool study guide that we've posted on Google Classroom. So check that out. That's going to have all the topics we're going to cover. Jenna, explain the difference between stroma and stomata for sure. So stroma is a part of the chloroplast, and it's where the Calvin cycle takes place. Stomata is an opening and many openings in the leaf where gas exchange takes place. Remember, CO2 comes in, O2 leaves, that's stomata. All right, another name for a stomata could be stoma. But when we say stroma with an R, that's a part of the chloroplast. All right. What else? Oh, I lost you. Let me scroll. This thing gets stuck sometimes. It's weird. What are the similarities and differences between ATP and glucose, Karthi? ATP is energy, basically. Glucose, you would have to like break up glucose. Good question. You'd need to convert glucose basically into energy. So glucose is food, right? It's a food molecule, the smallest food molecule. You need to break that up and convert it to get glucose, to get ATP, excuse me. So ATP is the actual form of energy that we use, whereas glucose is, like when you think about what do plants do? In photosynthesis, they make glucose. And then in cell respiration, they convert the glucose into ATP. For us humans, we eat our food and then we convert that food into ATP. Okay. Ronit, why do plants in the jungle tend to grow so fast? They have a lot of what they need. You think about what kind of plants there are. They have a lot of sunlight. They get a lot of water. So there aren't that many limiting factors, but sunlight can be, depending on how tall everything is. Is transpiration only for plants? Yes, Stefan. Yes, it is. Will the test include apoptosis? I think it's just important, Ronit, to know that apoptosis is when a cell decides to eliminate itself. Now we used to use the wording with like a cell commits suicide, and I prefer not to use that really because Yeah, in a sense it is, but I want to use different wording. So it's when a cell has basically decided that it needs to get rid of itself, and sometimes the cell recognizes that if it doesn't get rid of itself, it's going to be damaging the cells around it. Self-preservation. It's not preserving itself though. It's actually like getting rid of itself. Yeah. It's preserving the rest of the environment. That's true. My wife's helping me with my wording. So basically what a cell does is it signals for the lysosomes to open up, and they spew their enzymes everywhere, and that basically gets rid of the cell, eliminates the cell. So that's what apoptosis is. Okay, let's see. Shreya, limiting factors for photosynthesis. So think about all the reactants. What does it need? Right? When you think about the reactants, it needs water. It needs CO2. It needs sunlight. So any of those, it has to have all of those. Surya, why do cacti grow so slowly? Think about the same thing. Are they getting everything they need? Everything a plant needs? So in the desert, like what's the limiting factor in the desert? I think you got this one. What page is the plant leaf diagram on in the textbook? I do not remember that, Stefan. I don't have a textbook on me, so somebody else will have to help you out with that one. Hi, Roma. Why does a cactus grow so slowly in the desert? Yeah, so Roma, same thing. Think about limiting factors. Think about what it doesn't have. And let me know, and I can explain it further, but I think you can go from there. Let's see. What is the difference between ATP and glucose in terms of energy storage? ATP is a form of energy. Glucose has to be converted to become ATP, I guess, in a way. Roma, I think it's because deserts are dry and barely. Yeah, Gandhar, thank you. Plants need water to grow. Yep. John, what are the limiting factors for plant growth in the desert? Yep. So we just talked about this one. Where is the energy in the glucose stored? Let's see. So I'm going to explain it this way, Stefan. When you like think about cell respiration in the beginning, right, with glycolysis. When in the beginning, when you break up glucose, what does that take? It takes it takes energy to break something up. So it takes ATP. But the mitochondria is going to make money by bringing glucose in. So don't dive too deep into it in an AP bio level, because I don't want to lead you down that road. I want to make sure we keep it sort of at the basic bio level. So some of the information I'm going to give you may not necessarily sound like I'm putting I'm tying all the knots together to have it make sense. And the reason is because I think I have to be careful about not diving down that road of like, now here we are talking sort of AP bio. So if there are questions that I can help you with on the side, I'm happy to go that direction too. And we can just do that on the side later. Okay, let's see. Anika, good question. So how do plants make their stomata open and close? So I think there are two things we got to talk about here. Number one, I need everybody to understand rate of transpiration and what that causes with the stomata opening and closing. So if you have a plant that's in a hot and dry environment, the amount of water you have inside the plant is more than what you have outside the plant. Where do you have a higher concentration of water? You have more inside the plant than you have outside the plant. So what happens? Water is going to want to go from high to low. So water goes from high to low, which means from inside to outside, which means the rate of transpiration is high. The ROT, the rate of transpiration is high. When the rate of transpiration is high, the plant has to basically conserve how much water it has. So at some point, it's going to close its stomata. Okay? Stomata are going to close. Now, if the plant was in a, say, like humid environment, now you might have more water outside the plant than you do inside the plant. So what does that do? That means water wants to go from outside to inside. The rate of transpiration is low, so the stomata open. It's open, it's closed. The other thing I want to make sure you know is that this opening right here, that is called the stomata. Okay? This guy and this guy, these are the guard cells. So the way that guard cells work, remember this is closed, this is open. So you have ions, say like potassium ions. that rush into the guard cells. And as they rush in, water follows via osmosis. Okay, and that plumps them up. That plumps up the guard cells, and as they get plump, they start to kind of look like this. So they look like they're opening up, and they're opening that window to the stomata. Now, when the ions end up leaving, the water ends up following, and that kind of makes them skinny, and that makes them lay flat against each other, therefore closing the stomata. I hope that explanation made sense. That covers kind of what I need you to know regarding leaves and stomata. Okay, let me try to find my place again to see what questions. Yeah. Gondar, I love that you're explaining more. What is the difference between ATP and glucose? Oh, I saw that one already. What are the differences between chromatin, chromatids, and chromosomes? Okay. I guess we can just move on then, huh? Are we good with photosynthesis? I guess we are. We'll just move on. Yeah. Functions. So, Jenna, chromatin is the jumbled version of DNA. So it's just all jumbled up. Chromosomes, I think I'm going to draw. So I'm glad I got this. Some of you, so this is something that we talked about in class, and I want to make sure that you understand this. So this right here is a chromosome, okay? This middle guy right there, that's a centromere. This whole thing is a chromosome. This right here, this jumbled up stuff, that's chromatin. There's no really structure to it. So that's like spaghetti style DNA. That's chromatin. This is a chromosome, okay? When the chromosome replicates, what it does is replication means it has an identical version of itself, but they stay like that. They kind of share that centromere, it looks like. That's what it looks like, okay? Now, this is still a chromosome. It's a replicated chromosome. However, it's got two sister chromatids. So here's sister chromatid 1, here's sister chromatid 2. They're identical versions of each other. So this is still a chromosome, but it looks like a double chromosome. It's just basically a replicated version of a chromosome. Okay? So what I drew originally, that was a chromosome. This is also a chromosome. It's just a replicated version, and this is chromatin. This is the jumbled up version of DNA. All right. I hope that helps, Jenna. All right, let's keep going. Surya, I'm going to come back to your question. I know that I keep skipping it. I want to come back to it because I want to give you a correct answer because it might take me a while to explain that one. What is the function of spongy mesophyll? We'll come back to those. So I'm going to come back to the leaf questions in just a little bit. Plants usually green, so because the chlorophyll that they have, Surya, that's why. John Winton isn't a pigment. So as usual, I love that you're all helping each other out. Nice job. Keep that going. Yep, Gandhar, you're right. The pigment chlorophyll absorbs other colors and reflects green, I think. Yep. Yep. Nitin, what signals the ions water to go into the guard cells? Is it just from the environment? I don't know. I can find out. I can find out and look that up for you, but I don't know like what's signaling that. I gotta look that up. Let's see. Why do chromosomes condense before being separated? They need to condense because like if they try to divide up when they're in the shape of spaghetti, there's no rhyme or reason to it. You need to make sure that they are in a sort of a more structured form before they actually start to replicate so that you get, it's very calculated. You have to do it perfectly. It has to be done perfectly. Otherwise you can't just kind of guesstimate like where half of it is. That's why they condense. Yeah, Ishita, it's so the DNA replicates properly. Why isn't the energy stored in glucose transferred to carbon dioxide and water? Let's see, John. Why isn't the energy in glucose stored in glucose transferred to CO2 and water? The products of cell respiration. I'm having a hard time understanding this question, John. See if you can ask me in a different way. Okay, Stefan, stomata are only found on the underside of the leaf, and the palisade mesophyll is only found on top. Yes. Yes. Stomata aren't only found on the underside. There are some stomata on the upper side of the leaf as well. How does surface area affect diffusion rate? Percent surface area. So this is I was having a really good conversation with Jenna today after school, and we were talking about how like compare a tiny ping pong ball to a soccer ball and then try to make that even bigger and make it like a beach ball. Each time it gets bigger, both surface area and volume are increasing. However, volume increases at a faster rate than surface area, which is why cells can't get too big. Okay. Hope that's helpful. How do environmental conditions in the rainforest allow plants to have a high rate of photosynthesis? Because typically you have a lot of water. Typically, in a rainforest, those plants are pretty much getting what they need. They have all the reactants to make photosynthesis happen. Yeah, Como, hi. DNA overload. So DNA overload, this is kind of how I look at it for those of you who whose brains work with analogies. So like in a classroom, imagine if I was DNA. So I'm coding for proteins. I'm coding for lots of different stuff. I'm sort of in charge. And I can handle, say, like, I don't know, like 36 to 40 students, right? And all of a sudden, we're going to try to make the classroom bigger, like twice the size, and we're going to add lots more students. So all of a sudden, we have like 80 students or 100 students. At some point, I'm going to be overwhelmed or overloaded. The DNA can only handle coding for a certain amount of proteins within that cell, and you want to make sure you don't overload or overwhelm the DNA. Roma, how do limiting factors affect diffusion? How do limiting factors affect diffusion? I guess I need to know like more context. In what sense are we talking about? Energy in cell respiration starts out in glucose, then moves all the way to ATP, okay? Why doesn't some energy go to the products of photosynthesis, like some carbon dioxide and water? Well, you have to remember that there's still energy being used along the way. Like some of the things that we do, like breaking up glucose, that requires energy too, right? Yeah, that's how I would answer that question. Okay, let's see. So Archit, the plant's green was answered already. Is a telomerase enzyme located somewhere specific? Nika, hi Nika. Nika, I don't know where exactly it would be placed, that enzyme. But regarding telomerase, we know that telomeres are basically the caps, right? The cap. Remember those, those pens that have a cap to it, or like one of these guys that has a little cap to it. So this is how I see, if I can open it, this is how I see telomeres or telomeres. Wow, I just noticed there's an eraser at the end of it. I didn't know that. Each time a cell divides, these telomeres get shorter. And when you look at the enzyme telomerase, when you look at cancer cells, there's a lot of telomerase in there because they're not allowing their telomeres to get shorter, which is kind of crazy. Chlorophyll is a pigment. Yep. How does... Shreya, oncogenes, we didn't get that far, so Ms. Detmer and I are gonna dive back into cancer the week before final exams. So yeah, we didn't really get to talk about it a whole lot, which bummed me out, but we're gonna come back and teach it again in a couple weeks. Nitin, how does the higher ratio of surface area to volume hurt a cell? It doesn't hurt a cell. It basically means that, so look at it this way, again, going back to an analogy. So surface area to volume, think of it this way. Surface area is like the amount of streets, roads, and bridges that go into a city, and volume is the size of that city itself. So the issue is, as the size of the city gets bigger, the surface area or the roads should get as big, right? Not as big, but as many roads and bridges into that city. The problem is that mathematically, when a sphere is getting bigger, volume is increasing at a faster rate than surface area. And that means that city is getting much bigger and it doesn't have a whole lot of roads to get to that city. So if something tries to get in, yeah, it can still get in because you still have a phospholipid membrane or bilipid membrane. But once it gets in, it has to travel a really far distance to get to where it needs to go. So again, go back to that ping pong ball size versus sort of a beach ball size. It's kind of what we're talking about. As a gene, a segment of DNA that codes for a specific property. Stefan, you are 1 unit ahead, my friend. But yes, a gene is going to be a segment of DNA that codes for something specific, like a specific trait. Yeah. John, why is interphase the longest phase? Because I don't know. I don't know why it's the longest phase. A cell spends most of its time in interphase, which is made up of G1 or growth 1, S phase, also known as synthesis phase. That's where DNA replication happens. And then it goes into G2, growth 2, and then it goes into M phase, also known as mitosis. Remember, you don't need to know the specific steps of mitosis anymore. Back in the day, we had to teach these, we had to learn these, where you'd go, okay, so it's prophase, anaphase, and so on, metaphase, telophase. You don't have to know those phases anymore. That's kind of a thing in the past. It's no longer in California standards. Let's see. All right, I'm going to see if I can catch my place again, and I'm going to see. I try to help you out with other questions that are coming up. Okay, let's see, where were we at? So Ishida, yep. Roma, for one of the A-level questions, when a cactus is in the desert, when it has limiting factors, how do we include diffusion in our answer? Oh, okay, I see where you're going with this. Okay, I finally understand. Thank you for explaining it that way. So you have to think about it this way. Diffusion is going to have to do with sort of like what's going in and out. So think about if it has stomata, right? Is water going to want to go in or out? Are other materials going to want to go in or out of the stomata? And so if there's a lot of water inside versus a lot of water outside. So that's my hint to you. So diffusion / osmosis is going to have to do with kind of where's everything going. In regards to the temperature, in regards to how much water there's inside the cell and how much there's outside the cell of the plant. Let me know if that helps. If it does, awesome. If you're still confused by it, we can keep working on it. Okay, let's see. So regarding the palisade and the mesophyll. So basically palisade, they're known as like palisade mesophyll cells. They're pretty tall. And they're supposed to absorb a lot of light. Okay, they have lots of chloroplast. Now, spongy mesophyll cells, those have a lot of air spaces between them, and the point of that is for them to basically have a lot of gas exchange. So one of them is tall. The point is, like, can we pack just a lot of chlorophyll in there? The other one is just sort of like there's a lot of air gaps in there. I hope that's really all I want you to know. More than that would be unnecessary. Nitin, is DNA overload more a single cell thing, or does it happen when there are many cells? DNA overload is the idea of, it's a concept, it's an idea. It's not something that actually really happens. It's an idea that you could overload DNA by trying to increase the cell size. So it doesn't make sense to increase the cell size if it's going to put a lot of demands on the DNA, and that's why cells don't get that big. That's why the human body is made up of so many cells instead of like 100 cells that are just larger. That's one of the reasons why. How are carbohydrates related to respiration? Archit, carbohydrates related to respiration. I guess you can look at it as like when carbohydrates are broken down into glucose, that glucose will help you with cell respiration because glucose is one of the reactants. Yeah, Ishida, you answered the question for me. I should have just looked at what you said. Do pyruvic acid or acetyl-CoA have more energy than glucose? And if not, why? Well, they're smaller, right? So pyruvic acid and acetyl-CoA. Pyruvic acid is a 3-carbon molecule. Acetyl-CoA is a 2-carbon molecule. So they're not going to have the potential energy that glucose has. Okay, but it's a process. Glucose needs to be broken down into those two things, and it needs to go through the Krebs cycle or the citric acid cycle in order for them to finally get to the ETC where a lot of ATP is made. All right. Let's see where you're all at. I think I fell behind big time. Surya, what substances move inside and outside of the stomata? So CO2 can go in, water can go out, but remember, they're all in the form of gas. Cellulose is made up of glucose, and it creates strong cell walls in plants. That is true, Gandhar. All right, you're all helping each other out, so I'm going to skip past those. Does darkness, Komal, there's a question from you. Does darkness affect whether stomata will be open or closed? Not so much, but darkness is going to affect the light-dependent reactions. What is the point of cells dividing other than allowing the organism to grow? Why does the organism need to grow? Good question. Okay, so Nika, you're asking a really good question. So big picture, an organism needs to grow because your DNA is coding for proteins to be made. So for example, for humans, if your DNA is coding for you to get taller, that's just what's going to happen. In order for you to get taller, you're going to need more cells. So there's constantly division in that sense. You also might have injuries throughout your lifetime. Every time you're injured, your cells are going to try to replicate so that you have more cells. So that's another reason why mitosis is constantly taking place. And just remember too that a lot of our cells are constantly getting replaced. Like you think about your skin cells and you're constantly replacing them. You think about your red blood cells. On average, they're around for 4 months before they get replaced. I hope that helps, Nika. Shreya, P53 is a protein that controls the gene expression of other cells, and cyclins control. Yes. Okay, so let's talk about P53 and cyclins. Okay, another analogy. P53 is like the sheriff in town. Cyclins are the officers that the sheriff has hired and trained, right? So when cancer comes in, so if you're cancer and you're trying to really create chaos, would you go after cyclins or would you go after P53? Hopefully your answer is P53. Because if you get rid of P53, then you also, in a way, are kind of indirectly getting rid of cyclins, because P53 isn't there to code for cyclins anymore. And cyclins regulate the cell cycle. So if something's going on, cyclins are going to catch that and say, hey, this is a no-go. And the cyclins are going to send a message to your cell being like, hey, we need to go through apoptosis. So if P53 is no longer coding for cyclins, no longer telling cyclins what to do, cyclins are basically useless, right? And so the cell is going to be able to divide in any way that it wants to, which is cancer. Okay, does chromatin make up chromosomes, Ria? So Ria, chromatin and chromosomes are the same. It's just uncondensed versus condensed versions. Chromatin would be uncondensed, chromosomes would be condensed. Yes, keep helping each other out. This is helping me out too. Thank you for answering each other's questions. Nitin, explain cell respiration. Nitin, be more specific. What do you mean? Do you want big picture? Do you want me to walk you through Eddie, the electron? What is it that I can help with? Stefan, is the energy in glucose transferred to other molecules? Yes, it is going to be transferred. That energy is constantly transferred. Cancer cells keep dividing because they have a defect in which keeps them. Yep, you're right. So does anyone know what negative pressure is? Lavi, you don't need to know what negative pressure is, at least for my class. But I know you're not in my class. All right. I love that you're answering each other's questions. Hi, Minsu. The volume to surface area ratio problem. If we were to explain the problem, could we use math? Yeah, for sure. You can definitely use math to explain the surface area to volume issue. Just remember, volume grows at a faster rate than surface area, and that's a problem. Why do cells go through apoptosis, Lavi? Because something has gone wrong. Something has gone wrong in the cell. And the cell, it's sort of like a noble act, right? The cell's like, something's wrong and I'm going to get rid of myself. So that's what apoptosis is. Will windy weather cause ROT to increase or decrease? So if it's windy, that wind is going to push all of the humidity away from the leaf, which means that the ROT, the rate of transpiration, is going to be high, which means that the stomata are going to close. Let's see. Yeah, Hira, you don't need to know source and sink because we didn't. I would have liked to spend more time on that, but we didn't. So I didn't put any of it on the test. Yeah, 57 people watching. Good stuff. Yeah, Nitin, you want LED electron? Okay. So Nitin, one thing I want to also recommend, in addition to me telling you the story, is make sure you go in Google Classroom and just read up on that story. I think it'll help just reading it too, visually seeing it. But I will teach you LED electron again. So and remember, in the beginning, you have glucose, right? And I'll teach you the sort of realistic version instead of the kind of fun and goofy Eddie version. So you have glucose. It's hanging out in the cytoplasm. Glucose is going to be broken up. You're going to spend 2 ATP to break up glucose into 2 pyruvic acids, okay? And each pyruvic acid is then going to have to pay 2 ATP to get inside. So that's glycolysis. Glycolysis is the breakup of glucose because it's glycolysis, glucose breaking up. That happens in the cytoplasm. So pyruvic acid is going to enter the mitochondria. It then is going to break up into acetyl-CoA. Acetyl-CoA is a 2-carbon molecule, whereas pyruvic acid is a 3-carbon molecule. The reason they're breaking up is because they need to join the citric acid cycle or the Krebs cycle, which has 6. Citric acid has 6 carbons, but at the moment, there are already 4 carbons sitting there, so there's room for 2. So pyruvic acid breaks up into acetyl-CoA, and now you have citric acid, which is a 6-carbon molecule. Again, citric acid is going to break up again, release a lot of electrons, and then those electrons are going to be high energy. They're going to go through the electron transport chain, and that's going to create a ton of ATP. Remember, in both photosynthesis and cell respiration, the ETC, the electron transport chain, is where you get most of your ATP. How is cellulose related to photosynthesis? Cellulose makes up plant cell walls, right? And photosynthesis is done through plant cells. Where is glucose stored in plants, Stefan? Good question. Okay, so that glucose is going to be made in the leaves, right? And then from there, it's going to go to wherever it's needed. And that's sort of where the sink is. So where is it needed? So for example, these grapes, these uvas that I'm eating, they have a lot of glucose. So the grape leaves are going to be doing a lot of the hard work in making the glucose. And then the glucose is going to get pushed to or get delivered to where it needs to go in the actual grapes itself. Hira, are there 2 chromosomes in a sister chromatid? Technically, no. So let's go back to this drawing. I mean, this is what makes this challenging, right? You look at this and you're like, that looks like 2 chromosomes. So in a sense, I guess you could say that, yeah, because ultimately these are going to separate. And when they separate, they're each going to be considered their own chromosomes. So I guess you can look at it that way. Just know that when they're together like this, we don't call them 2 chromosomes. We call them sister chromatids. But I think you already know that. Are only malignant tumors able to go through angiogenesis? So Shreya, I am going to cover malignant and benign tumors in 2 weeks. I haven't talked about it in class much, so I won't get into it right now, but I will definitely cover that in a couple of weeks. What has to happen in a cell for cancer to occur? So you could have you can have things like an outside factor like carcinogens. It could be anything. It could be UV rays. It could be radiation. It could be things that we choose. And remember, environmental factors means anything that happens to us because of our environment. Like that could even be your choice to drink alcohol. Alcohol is considered a carcinogen. So if it's not environmental, that means that it's hereditary or genetic. Difference between cancer cells and stem cells. Cancer cells are cells that have kind of gone crazy. Stem cells are cells that are unspecialized, so they can become any type of cell that we want them to. Do we need to know all 4 reasons of aging, Viba? I don't think so. Okay. Yep. Gondar, you're right about if the error is not caught, apoptosis won't be performed. Roma, for the A-level question about the cactus and limiting factors, how can we include CO2 in our answer? Can we just say that CO2 is a reactant for photosynthesis, or do we need to explain that more? No, you can just talk about how CO2 is a reactant. That's fine. Can you explain what happens to acetyl-CoA when it enters the Krebs cycle? So when acetyl-CoA, here's the thing. When acetyl-CoA enters the Krebs cycle, it's no longer considered acetyl-CoA, Shrika. At that point, it is considered citric acid, and citric acid has 6 carbons in it. Okay, I'm seeing some questions that I've answered, like plants growing in the jungle, so I'll skip those. You can go back and check out my answers. And there's a lot of phenomenal answers here that you're giving each other. Hello, Serena. It's nice to see you. Let's see. I just lost my place. All right. Yes. Okay, one thing I want to cover. I keep losing my place. But one thing I want to cover is fermentation. So remember, when oxygen doesn't show up, because oxygen is the final electron acceptor, when it does not show up at the end of cell respiration, that's when you go into fermentation. There are 2 types of fermentation. Remember this. This is very important. There's lactic acid fermentation, and there's alcohol fermentation. Lactic acid fermentation happens in muscle cells, so in animals. And the way that happens is you get pyruvic acid to turn into lactic acid, and that causes an NAD+, which allows electrons to be carried away. Okay? Alcoholic fermentation is what you would do to make something like wine or beer or whatever, right? It's when you have yeast in there and you're basically cutting off oxygen, and that's creating alcohol. All right? Remember this too, we talked about this in class. When you go into fermentation, at that point, it's considered anaerobic, which means that there's no more oxygen being delivered. Aerobic is when you have oxygen coming in. What are the possible environmental causes of cancer, Jasmita? So that's really anything that you do that's not hereditary. So the stuff that I was talking about just a couple minutes ago, that whether it's by choice or not, right? Whether you choose to smoke a cigarette or it's secondhand smoke, it doesn't matter. It's environmental. It's something that's not genetic or hereditary. How much do we need to know about cancer for the test? Not a whole lot, not Como, because I didn't really get into cancer a whole lot this time, but I will next time for our upcoming final. Ronit, mention question. If you review photosynthesis, cell respiration, hello questions, cell regulations for survival will be. I think so, Ronit. I think so, but I mean, there's a lot of details in there, right? So generally speaking, yeah, if you know these things, you'll be fine. So I know, Ishida, it's going by. If time flies when you're having a great time talking about bio. It's already 6:50. All right, let's see where else we're at. I see that you're helping each other out. Stefan, when you talk about oxygen as the final electron acceptor, are you talking about H2O? Okay, so oxygen is considered the final electron acceptor, okay? However, yes, technically speaking, yeah, H2O is sort of what ends up taking that electron in, but H2O is not known as the final electron acceptor. It's actually oxygen that's known as that. Yep. Oxygen is oxygen the final electron acceptor, or is water? Oxygen is the final electron acceptor, but oxygen becomes water. So Stefan, how does converting pyruvic acid into lactic acid to get NADH into NAD plus? Like, how does it? So basically, in order for glycolysis to continue, Stefan, you need to be able to take, when we talked about Edna and how Edna leaves with Eduardo. So in order for those electrons to leave, you need an electron carrier. And that's why you need NAD plus. So that's why it's important. What happens when cells in the brain undergo apoptosis? Cells anywhere can undergo apoptosis, Hopefully it's an area where you can continue to divide and get more cells. How is citric acid broken up again once it is formed? Nitin, we don't really talk about how it's broken up. The citric acid cycle, you basically end up just kind of getting it broken, and the electrons end up being high energy electrons that leave and go to the ETC. That's what you have to know. Arjun, yeah, Arjun, I have a tattoo on my arm, and it's basically an imperfect redwood tree, because I think that there's a lot of beauty in things that are imperfect. We're losing a lot of viewers. We're now at 49, Parth. That's okay. Even if I have 2 here, that's important to me. So whoever can stay can stay. If you can't stay, no worries. Can you explain the whole electron carrier thing in cell respiration? Yes. So cell respiration and photosynthesis, both of them, a lot of different parts of them, when electrons are sort of like spewed out, when you break a molecule and electrons end up leaving, you need electron carriers to take those electrons to specific places. And that's what NADPH in photosynthesis is. That's what NADH and FADH or FADH2 in cell respiration are. Viba, alcohol causing cancer. I don't know all the details of it, but I can look into that, especially when we get into cancer. I can talk about that much more. Why do most cancer cells have mutated versions of p53? And why do drug makers, oh man, you want to get into this, John? Why do drug makers focus effort on boosting levels of John, I totally agree with you. I think the focus should be on p53, but also you have to remember that drug companies make a lot of money from sick people. So sometimes I think to myself, like, yeah, is it possible for us to be able to solve by going a different route? I think so, but there's a lot of money in pharmaceuticals, and I think that's something you have to remember when you think about stuff like this. Let's see what else we got. Ishida, your guys' spelling is on point. That's funny. All right. I think I might have gotten caught up, unless I missed a ton of questions in there. Don't they just not respond? What do you mean, John? Doesn't who respond? What are 3 things that ATP can be used for in a cell? Hi, Shivali. ATP can be used for lots of different things. So, for example, active transport could be one of them, right? What else? Help me out. Tell me what other things you can use ATP for. You can use it to break glucose, right? What else? Give me more. Give me more ideas. There's so many things. Who else? Just respond. What do we need to know about mitosis? Soft one, you just need to know the general, like generally what mitosis is. If I show you pictures of mitosis occurring, I won't need you to know which phase it's in, but I can point to the mitotic phase and be like, hey, how many chromosomes do you see in there? I can, what if you see like 4 chromosomes? Then I can ask you, or say you see like 6 chromosomes, then I can ask you when it replicates, when all these chromosomes have replicated, then how many would you see? Then you'd say 12. Right? Or I could show you a replicated chromosome and be like, how many chromosomes are there like before replication? Those are things you got to know. All right. Storing energy. So Nissa, I think we answered that question already about difference in ATP and glucose. So Ishida, why are half the cancer cells have mutated p53s? There's so much more I want to teach you all about cancer, and I can't wait to teach it to you. But I would say for the time being, you're all in a good place. But let's hold on to that conversation for a later time when we actually discuss cancer, because I would love to talk to you more about cancer. But it's hard when I haven't taught you some of the really important stuff about it. So Alex, we answered the cactus question already, so just back up and you'll see it. Ishida, what's confuzzling you? By the way, which period was it where I used where I made up a word and I said shrinkle? And also, how come people don't use that word more often? I think shrinkle should be a word. I don't even know what I was talking about in class when I said shrinkle. I think Gabby, if Gabby was here, she would tell me because somehow, so it was third period because Gabby remembers. All right. John, don't cancer cells just ignore p53 signals, or does p53 actually get mutated? They actually mutate p53. Yeah. What's the difference between pyruvic acid and lactic acid? They're different forms, they're different molecules. John, an A-level question asks, why drug makers focus efforts on boosting levels of p53? Their answer is supposed to be about financials. I would not ask you that question. Let's see. Third, yeah, soft one. Third period is where I said shrinkle. Ishida, you're confuzzling me now. I don't know what you're talking about. Yeah, Roma. Roma, how come you don't use the word shrinkle more often? Everyone should. You too, Ronit. Everybody who was in third should use that word shrinkle. Actually, everyone in general. Hi, Rudra. Rudra, we're still homies, even though you're not passing back papers from me for a while. I got a lot of heat for that one. Nissa, shrinkle? I don't quite know what shrinkle means. I was explaining something in class. Those of you who were there, you can explain it. And I somehow, I don't know why, the word shrinkle came out of my mouth. And then I stopped myself and I was like, that's not a word. But I think we all decided that it should be a word. Necrosis is basically when cell dies because of injuries and all that sort of stuff. Yeah, necrosis is when you have, Gandhar, thank you, when you have a group of cells or a tissue completely die. That's what necrosis is. Yeah, Lavi, you got it. Tissue death versus apoptosis, which is cell death. Exactly, Lavi. Explain. Somebody says, cyclins before the stream ends. So Arjun, I explained cyclins already. Just back up and you'll find it. Somebody help Arjun out and tell him where it was. But I talked about cyclins. Cells shrinkle when it's hypertonic. Is that what I said, Roma? A cell shrinkles when it's That's pretty funny. Apoptosis is when the cell actually kills itself. Yeah. Yep. Ronit shrinks. Parth, which period is my favorite? Parth, I don't know that I have a favorite period because they're all so different. So different. And so special and awesome in their own ways. You know? I get to have a little more experience with third because that's the class where Ms. Detmer gets to observe and kind of hang out and watch me. I have a little less experience with fourth and fifth since Ms. Detmer teaches those classes. So yeah. Fourth, Ishida. Oh, Ishida. I combined shrink and wrinkle. Oh, that's what happened? You're totally right. That's why I said shrinkle, because I combined shrink and wrinkle. Yes, I'm sorry to remember that. Alex, a cactus with a stomata. Yeah. Yes. If you use shrinkle in actual test format, will it be correct? You know what, Ronit? If you say shrinkle on the test, not only will it be correct, but I will also laugh. So, John, let's go back into the cancer cell, the p53 thing. Okay. So let's talk more about it. I don't have it in front of me right now, so if you can type it up for me, I can help you out with it a little further. Now I see where you're going with this. I see what you're saying. Alex, let me try to look this up. Because I have not looked up location of stomata on cactus. Okay, so check it out. The stomata of many cacti lie deep in the plant's tissues. This adaptation helps cacti reduce water loss by keeping the hot, dry wind from blowing directly across the stomata. The leaves and stems of many desert plants have a thick, waxy covering. I hope that is helpful. Gondar, I know we don't know each other very well because you're not my student, but I really appreciate having you on here, and you're awesome, and I wish that you were my student. Yeah, Alex, I hope that explanation of the cactus helped. It's fourth because we have the best paper passing time. Arjun, fourth does have the best paper passing time. This is true. Ishida, I like your sense of humor. Ishida, you need to talk to me more in class. That's me. Arch it. I like how there's a battle right now between periods. You know what my dad used to say to me? I'll tell you this. So I would sometimes ask my dad when I was a kid, because my dad had 5 boys, and I'd be like, Dad, who's your favorite? Right? And this is what he would say. He would hold up his hand like this, and he'd be like, my 5 sons are like my 5 fingers. I love all of them equally. That was my dad's response. So this is what I'll do to my bio periods. There's actually 4 of them, because I have 6th grade sheltered. I love you all equally. John, the A-level question is really long. John, how about this? When we're done, if you want, I can email you and we can chat via email. No problem. Or you know what? Let me try an even better idea. Let me try to look it up right now. Give me 1 second. Un segundo. Hold up. 1 sec. Okay. Let's see. A-level question. Oh, that's not. I am looking in the wrong place. Oh, man. Okay. So let's see. I'm looking at all the possibilities we gave you all. So the first one was the 6 friends eating pomegranates, right? The next one was 6 friends sharing a bowl of grapes, like I'm eating right now. And then the next one was what factors limit plant growth in the desert? Okay, we've talked about that one. Okay, number 4, this is what you're talking about. Okay, I'm going to read this out loud. Of the nearly 1.7 million, I can't talk, 1.7 million people diagnosed with cancer each year in the US alone, about half have mutated versions of p53. Yep, a sign of how important the normal gene is in preventing the disease. It is one of the most intensely studied genes in science and a highly sought after target for drug makers. But in dozens of p53 drugs in development, the vast majority simply try to boost levels of healthy p53. And despite the decades of effort, none has made it to market. Why do you think half of all cancer cells have mutated versions of p53 alone? Okay, so explain what p53 does. Please underline any academic biology terms using definition. Okay, so first of all, it's important to know what p53 does, which I know that you know already. So then I want you to really kind of go at it from cancer's perspective, right? So if you're a cancer cell and you're trying to divide and you're trying to make the cell go crazy, if that's your aim, if that's your goal, then you're going to go after p53 instead of the cyclins. That's how I want you to go about this. Why do you think drug makers focus their efforts on boosting levels of healthy p53 for cancer patients? Because if you have healthy p53, if you have a lot of that, that can help sort of, if you have healthy p53, it can't be, what's the word I'm looking for? Cancer cells can't come in and sort of like destroy p53 and sort of like take over and eliminate it. Infiltrate. That is the word I was looking for, which my wife helped me out with. Infiltrate is the word of the night. All right. It is not shrinkle. All right. Let's see. All right. So we're reaching the end of our time. So I'm going to see if I can go through any questions that I have not answered yet. Let's see. So John, I hope that was helpful. Oh man, you guys are funny. You are a funny bunch. So Como, food and cell respiration. So in order to do cell respiration, we need glucose. And that's how food and cell respiration are related, because glucose is a reactant. Stefan, can cyclins be replicated? How about p53? I don't know, Stefan. I don't know if cyclins and p53 can be replicated. How does the ratio? Okay, so rear-eye. I answered this one already, so just rewind and you'll find the answer. How does a cell become cancerous in the first place? So Arnav, it could be environmental factors. It could be lots of different things. But I mean, for what we know, you can talk about carcinogens, and that's how cancer is caused. Okay. Vibha, can P53 cause cancer? No. P53 is sort of like the sheriff in town who's supposed to prevent anything bad from happening. So it's not going to be the thing that causes cancer. A lack of a healthy P53 is going to cause cancer. So Stefan, because the cell performs photosynthesis and cell respiration, does it mean that it releases both oxygen and carbon dioxide? Stefan, technically speaking, yes. And I just bit my tongue. Technically speaking, yes. So oxygen is so a cell is going to, or a plant is going to release a lot of oxygen, but there's still a tiny amount of CO2 that it's going to release. Okay, let's see. We're about to end, so let's see. You don't need to know the 4 stages of cell division. That's not something we need to worry about at all. Nitin, do all the electrons lost throughout the process of CR go down to ATC? That's okay to think of it that way. Yeah. Just think of it as there's a ton of ATP that's made because of the ATC and electrons. Vibha, you want to see my dogs? Okay. We are going to be ending the, whatchamacallit now, our session. I want to remind you to eat healthy snacks like uvas, like grapes, and drink healthy things like pomegranate juice, which I'm doing. I'm going to call my dogs over so that you can see my dogs. Let's see. Coda, hi, buddy. You want to come over here? Kuchuk, you want to come over here? Come on. I have to get him a treat. So I'm going to get these guys a little treat. All right. You all want to see Kuchuk and Dakota eat an apple or eat apple wedges? All right. Let's see if I can show you all where these 2 puppies are. All right. You see them? You see Kuchuk and Dakota? Hi, guys. Can you sit? Yeah. Good puppies. Good puppies. Kuchuk, you guys want to see Kuchuk catch us in the air? She's awesome at it. Ready? Oh, missed. Caught. Here, buddy. Good boy. All right, there are the dogs. You happy now? They look hungry, Parth. Parth, these dogs act like they're hungry. They're not hungry. That's just what they do. They come begging for food anytime. Thank you to everybody. Mr. Jones, I see the Jonesy. Good to see you, Mr. Jones. See you mañana. Parth, you love uvas. No problem, Nissa. Anytime, everybody. It was good to see you all. Thank you for joining me. Who's who, Kandar? So the chocolate lab is Kuchuk, and the golden retriever is Dakota. Yeah. What did you say? Ishida, you're not responsible enough to have a dog according to your parents. Ishida, if you like dogs, I'll try to bring my dogs to school on a weekend or something. You can play with them. That way you can get your dog fix. Arjun, bring them to school. You'd be my favorite teacher. Arjun, I would be your favorite teacher if I brought my dogs to school. I'm offended that I'm not already. Just kidding. Just kidding. Serena, you want to name your dog Fidel after Castro? Okay. All right. Well, the cool thing is when you get a dog, you can name your dog whatever you want. You know? Thank you, Arjun. I appreciate the added touch at the end. You should have an after-school dog playing session. Yeah, that would be awesome, actually. It's just, at a public school, you're not supposed to have dogs on campus. So I think it'd be really awesome. Just imagine like how much more relaxing it would be if like there's a student who's just kind of having a bad day, and at lunchtime they come and play with dogs. How awesome would that be? I would dig it, but I know that not everyone's into dogs, which I understand. Actually, I don't understand, but I respect. Okay. Mr. Jones, there's a question from Mr. Jones. I don't know if he's still here watching, but there's a question for Senor Jones. Are you going to stream soon? I know Miss Chow is going to stream in like 20 minutes. It's 7:12. She's going to stream at 7:30. Is the Eddie the Electron story on Google Classroom? Yes, Jennifer, it is. Just look on there and you'll find it. And if there's anything that you all can't find, let me know and I can help you out. John, I know dogs are amazing. Just like saying you like crazy. That's true, Nitin. That would be funny. That's probably the reason why I would not name my dog Fidel Castro. Yeah, Gondar. Yes, you're right, Gondar. Thank you for helping Lavi out. I appreciate it. All right. I think that's it. Thank you all for being here, for watching. I hope everybody has a good night. I look forward to seeing you all tomorrow. Take care, everybody.